Hi, my name is Madison Duffy, and I'm a master's student in the Gross Lab. I am from Colorado. I'm from a little town that's next to Denver called Littleton. It's right in between Denver and the mountains, and it's the best. What inspired me most to become a scientist was spending a lot of time in nature as a kid. I just have always felt more of a natural sense of curiosity towards the natural world. And then when I came to college, it kind of translated into me going into the sciences. I chose to go into microbiology actually because I took an intro class for it. I originally started in nutrition and dietetics. My growing up, obviously in Colorado, I feel like it's a more health conscious state since everybody's always active. So it's just it seemed very natural to me to want to go in that nutrition or exercise science direction. But one of the requirements for that was taking MMBIO 221, just the intro to microbiology class, and then the 222 lab. And I loved it so much that on the second week of the semester, I changed my major and I switched to microbiology. So the project I'm working on right now is a project on a protein called ataxin-2. Ataxin-2 is responsible for stabilizing RNA as it goes to be translated. When your cell is exposed to some kind of a stressful condition, like if there's not enough sugar, if there's too many reactive oxygen species, some things causing damage, it will collect all of the RNA that's floating around in the cell and some proteins, and it'll form these balls. So it will stop those things from being translated or being activated, and it will save energy. We're looking at electronic health records that people have submitted to in a program called All of Us, which is linking electronic health records and diseases people have been diagnosed with their genome. So we're able to see what mutations people may have in this particular protein, what diseases it's caused them in their day-to-day life. And then we're taking those identified mutations, putting them back into human liver cells, and we're measuring how it's changing lipid production or fat production within those cells. So it's kind of a backwards way. Usually you figure out, a disease. This time we're looking at the disease, finding the mutation, and then trying to take it back to the cells and figure out exactly what's happening to cause this issue. My favorite part of being a scientist, I think it's just really cool how everybody is so collaborative. And I don't know if that's just a BYU thing or it feels like it's everybody else as well, but no one person's doing an experiment or discovering something that's entirely built on their own work. You are always using something that was made by someone else or discovered by someone else. So everything is being built on the shoulders of other people. The hard work that one person put in and what for them was important, but for others, maybe it wasn't. And now it's foundational for what you're doing.